Okay, so we've made all of this glucose. That was our first step. We captured the energy from the sun to by plants, and we used carbon dioxide and water. Those are our inputs. We also had sunlight. We also had some enzymes in there. We produce oxygen that we breathe. This is a byproduct. And then we produce glucose. This is where the energy is now stored in the chemical bonds of that molecule. So now what happens? Well, we try to assemble the glucose into larger structures for longer term storage. So the glucose is used directly by the plant for energy, right? It's a process called cellular respiration, which produces ATP that we talk about a little bit more later. So what doesn't get used, though, gets stored. So it gets stored in the roots of the plant for a later time. So when there might not be as much sun, water, or carbon dioxide, all the things that's needed for photosynthesis, you have the plant has some glucose molecules that it can draw upon at any time. So before it can get stored, though, the glucose has to get repackaged. So all of these glucose molecules get linked together in these long, long chains, which we call carbohydrates or polysaccharides or starch. They also get assembled into cellulose, which we don't talk about too much. This is part of like a structural component of the plant cells as well. But it gets assembled into these long storage molecules called starches or complex carbohydrates or polysaccharides. Those are all basically synonyms for long chains of single glucose molecules or single monosaccharides. These are a more stable energy storage molecule. They can save energy for long periods of time. Okay, just to do a little bit of chemistry here, organic means that a substance has to contain both carbon and hydrogen. So some examples we've talked about in class are glucose, starch, carbohydrates, proteins, amino acids, nucleic acids. All of those things contain carbon and hydrogen. Remember cho-cho, chon, chomp. Things like water, which is H2O, doesn't contain carbon, right? Carbon dioxide, which is CO2, doesn't contain hydrogen. So those are inorganic molecules. So when a big molecule is made from a bunch of smaller molecules, like we're making starch from a bunch of sugars, the process is called dehydration synthesis. You don't really need to know a lot about this except for really just the name. Synthesis means to make or to put things together. So that's an easier way to remember this process. When we break up a molecule into smaller pieces, so when we take this starch and we break it back down again, it's called hydrolysis or digestion. Forget this slide, don't even look at it. Okay, dehydration synthesis. When smaller molecules are assembled, see this bond that's formed here? You have one monosaccharide and another monosaccharide. And through this process, we're going to lose a little water. That's where the dehydration comes from. And we're going to produce this chemical bond between those two monosaccharides that create a chain. This is a sucrose molecule. It's just two simple sugars together. But in starch, this would be a long, long chain of them. Just a little fun fact there. Usually when a chemical ends in the letters OSE, it's probably a sugar. All right, how come we're not going? Here we go. Hold on. What just happened? Okay. So just like sugars, other biological molecules are synthesized in the same way. Fats, which we also call lipids, are made up of fatty acids and glycerol. Proteins are made up of amino acids. Nucleic acids are made of nucleotides. This is I like this video a lot. I would take a few minutes to watch this, maybe take some notes on it. And all of this information is summarized in this chart here. So in this case, we're looking at the monomer would be monosaccharides or glucose, polymers, what's in this thing, and then what it does, an example, and what elements are in there. You can see all of these contain carbon and hydrogen. That means they are all what? Organic or inorganic? If it contains carbon and hydrogen, it's going to be organic. Amino acids are unique because they contain nitrogen, and nucleotides are unique because they contain nitrogen and phosphorus. All right, indicators. You should know that iodine detects starch. Benedict's detects sugar. You don't really need to know burettes. You don't need to know the fat, but we did do those. They're kind of fun, but you don't need to know those for the test. All right, and so now we've got all of these long storage molecules of carbohydrates, also proteins and fats, and now what happens? We digest them. There are two types of digestion: mechanical, which is like breaking it up, also peristaltis in your esophagus, and then chemical digestion. This is the most important. This is where our body uses acid, which means it has a low pH, and enzymes, which are proteins that have specific biological functions. And they break up those food back into the individual components. So why do we have to digest them? Look how big this starch molecule is. We have 1, 2, 3, 4, 5, 6 simple sugars, right? That's a big molecule. But here's just one simple sugar, glucose. It's small. So we have to break them down because they are too big to easily pass into the cells. So we have to break them down by hydrolysis, so they can more easily pass through the cell membranes. And we did a whole lab on that. Don't watch that one again. It was gross. Okay. So chemical digestion, enzymes are used like amylase, which is used to break up starches. Enzymes are used to digest molecules back into their simple building blocks. Enzymes are proteins, often referred to as a biological catalyst, that speed up chemical reactions and are found in all living organisms because they are important in everyday functions like digestion and blood clotting and a million other things. They make your DNA, they help fight disease. They also act like scissors and cleave bonds between molecules. Some of them, every single one has a very specific function, but they all accelerate chemical reactions, and this is referred to as catalysis. So they and because they take part in a chemical reaction, but they are not destroyed in the process. So for example, see this enzyme? This is our enzyme here, and this is our substrate. The substrate is the thing that the enzyme is working on. We know that the enzyme and the substrate have to fit together like a lock and a key, okay? But this enzyme, so for example, let's see what's happening in this. The substrate is going to bind into the enzyme, and then this enzyme is going to cut this substrate in two and release the products. Okay, but this enzyme can get used over and over and over again before it eventually stops working. So enzymes are reusable, but they only work on something that's a very specific shape and size. This is also a good video. I like that. Take a second to watch that one. All right. So we know they're proteins. They are catalysts, biological catalysts specifically, and they speed up a chemical reaction. Now my slides are not advancing. And as I just mentioned, they work like a lock and a key. They have a specific shape that will only fit one substance, one substrate that it'll work on. So an enzyme that breaks down carbohydrates is not going to break down proteins, and it's not going to break down nucleic acids, and it's not going to break down, any waste products, because it only works on one specific thing. Okay, and we also talked, I'm going to just kind of skip through this. Okay. Oh, how cute is that? All right, so importantly, there are two main things that can affect the rate of an enzyme, how fast it works. The temperature. So as the temperature rate increases up to a certain point and then decreases because the protein changes shape and denatures. So let's take, for example, here's a human enzyme. We're seeing this is working best at 37 degrees because that's the same as 98.6 in Fahrenheit, right? 98.6 in Fahrenheit is about the same as 37 Celsius. So that's where you're going to see most human enzymes working the best. This one has very little activity when it's colder than that, increases activity to 37, then it starts to get too hot. It denatures, which means it changes shapes, and then it stops working completely. However, this enzyme is for a heat-tolerant bacteria. This bacteria loves the hot weather and lives in the hot environment. Its optimal temperature is something like 78 degrees. So at 20 degrees, it has no activity at all. Starts to get a little bit of activity at 40. Its optimal temperature is 78 degrees Fahrenheit, Celsius. Then when it gets too hot, it denatures, changes shape, doesn't fit the substrate anymore, and completely stops working. Same goes for pH. Although this is not really like as changing shape as dramatically as you get with heat, but the pH does affect the rate of reaction. So pepsin, this is actually an enzyme that works in your stomach. So it likes a very low pH. It likes acidity. And this is for trypsin, which I believe is in your pancreas. And it likes a basic environment. It's more like a little like 8.2. So just like temperature, enzymes have an optimal pH that they work best in depending on where they are located in the environment. Alrighty. So I think we are going to move on to digestion. All of this food, you just ate a big meal, your body is breaking down the proteins, it's breaking down the carbohydrates, it's breaking down the fats, it's breaking down everything into this nutrient soup, back into the individual building blocks so that your body can reassemble them in any way that it wants. So nutrients get absorbed through your small intestine, which is like right about there. So they get digested in your stomach. Well, digestion starts in your mouth, right? And then digestion, it continues to occur through your esophagus and your stomach, where it encounters that very, very low pH. And then the nutrients move into your small intestine. This is where your blood picks up nutrients that you have just digested. So then they get circulated all throughout your body, from your toes up to your brain, to provide nutrients and oxygen for every part of your body. Cell membranes, just briefly, these surround every single cell, and some molecules are small enough to go right in, like things you might have just eaten can diffuse through the cells, and some cannot. So this is where we get into transport, how we get things from your bloodstream into the cells of, let's say, your brain or your muscles, or someplace that they're really, really needed. We have two types of transport. One is active passive transport. So passive transport, this is for small molecules. Small molecules are tiny. They can just diffuse right through the membrane. So if you look at this little GIF, what do we say? It's a GIF or a GIF? Anyway, if we look at this, small molecules, this is the cell membrane. Small molecules going from a high concentration to a low concentration, like the top of the hill, the high part of the hill to the low part of the hill. They can just roll down. That doesn't require any energy. So going from a high concentration to a low concentration requires no energy. This is usually how passive transport is usually also known as diffusion, and small molecules like glucose, water, and gases pass through very easily. So they are usually, when going from high concentration to low concentration, moving by diffusion or passive transport. Osmosis is this phenomenon when we're just talking about water, and diffusion is usually referred to as other molecules. You can say diffusion of water. You can say that water diffuses too. It's just more specific to say osmosis, but diffusion is an acceptable answer. All right, and here's just a little, I'm not going to even talk about that, but read through those definitions. Active transport is different. If it's a large molecule, like that guy on the bike, right? If it's a large molecule, or you're going from a low concentration to a high concentration, you need a lot of energy to get up that hill. So going from a low concentration to a high concentration requires energy in the form of ATP. So to get across the cell membrane, if the concentration is low on one side and you want to go to the area where it's high on the other side, you're going to need energy or some sort of transport protein or something. All right, this is just kind of summarizes everything. Okay, last but not least, cellular respiration. Our energy up to this point has been stored in storage molecules like glucose and starch. Respiration is the process by which energy is released from the nutrient molecules and converted into usable form in cells. Okay, so what does that mean? Sugars like nutrient molecules like sugars have diffused through the cell membrane, but they can't be used directly by the cells as glucose. So they have to be converted to ATP, which can be used as an energy source. ATP stands for adenosine triphosphate. It is used in every single process in your cell. Everything that requires energy is going to use ATP. Glucose is a stored form. ATP is also a stored form, but it's just a form that is much easier for your cells to use whenever they need it. This happens in the powerhouse of the cell, the mitochondria. And this is kind of a typical picture of what you would see for mitochondria. And there are two types of cellular respiration, so two ways that we can break down glucose to make ATP. Aerobic, this is by far the one that we're going to be referring to the most. This uses oxygen. It makes a ton of ATP. It makes 36 ATPs from just one glucose molecule. Very efficient and used by organisms like humans, multicellular plants, and some protists. Carbon dioxide is a waste product. Anaerobic, however, does not use oxygen. This is when cells are starved of oxygen, or there are some organisms that just use this method of respiration regardless. You only make 2 ATP, which is not a lot, so it's considered to be much less efficient. And these are the kind of guys that use it. Waste products can also include lactic acid or alcohol. So lactic acid is that burning you feel in your muscles after you exercise and oxygen isn't getting to your muscle cells fast enough. That burn is the buildup of lactic acid. Alcohol is when during the fermentation process, it's anaerobic. Alcohol can also be produced. This is a summary of the chemical formula for cellular respiration, and what you can see is we're using glucose and oxygen, right? This is what you eat, this is what you breathe, this is what you drink, and we are breaking up that glucose to release the energy in the form of ATP, and we're also producing carbon dioxide and water. You breathe out carbon dioxide, right? You also breathe out water vapor. So this is the chemical reaction for cellular respiration. You should be fairly familiar with it because it's really just photosynthesis in reverse. Don't worry about that. This is a great little summary chart. Where it occurs, when it happens, what it needs to go in, what it needs to go out, source of energy. So in photosynthesis, the energy comes from the light. In respiration, it comes from the chemical bonds of glucose. The resulting, I love this one. Okay, so definitely take a look at that. Oh my gosh, we're getting close. That was just something I did for fun. We don't need to go into that right now. I love this little picture. Photosynthesis is where we make glucose. We store the energy right in those bonds. And then cellular respiration, we break it up and we release the energy to be used by your cells. And this is just showing you how the reactants for cellular respiration produce the products for cellular respiration. But if you notice, the products for cellular respiration are the exact same as the reactants for photosynthesis. And the products for photosynthesis are the same as the reactants for cellular respiration. So in plants, we've got carbon dioxide and water being absorbed, and they are producing glucose and oxygen, which we eat and we breathe. And when we eat and we breathe those things, then our body can break them down to make our energy, our ATP energy that we need. Okay. Look at that. Just an important thing to remember, all cells respire. They all use respiration, including plants. Don't worry about that. Okay, so I'm going to stop there. All right, I'll see you guys all soon. Bye.